record. Okay, so I want to welcome everyone who is here on my computer screen to this first Horizons lecture of the year. We're still putting together our Horizon series, but after last year ended abruptly, I really wanted to have Erin McKenney back, or well, here to start us off, because she had such an interesting background and her material was so compelling to me that I was really glad. And by then I felt like I had known her, so it's like, please come on. And so she was very willing to do that. So I really appreciate that. Thank you, Erin. So as an introduction, I just wanted to tell you how I was able to track down Erin and become familiar with her work. I was listening to a Fresh Air show, this was, gosh, probably a year and a half ago, and Rob Dunn, her PhD mentor, was being interviewed by Terry Gross, and he was talking about his book, Never Home Alone, I believe was the title of that, and it was fascinating. So I looked him up, and he directed me to you, Erin. And so it sounds like he's got a really interesting lab, but it sounds like you have done a lot more than just what you did in his lab. So I'm looking forward to hearing all that. Let's see. I don't think I need to say anything else, except I need to make sure that the chat is working. And so if anyone has any questions, let's see. I'm going to find this chat. There we go. Okay, so if anyone has any questions, I suggest you use the chat and keep your mics off on your computer. So I think with that, I am going to welcome everybody on this. And then I am going to figure out this. Where did my thing go? Can you still share, Erin?
You will need to stop sharing your screen before I can start sharing mine.
Right.
So there should be like a red stop share button toward the top.
Here we go. Okay, it had disappeared. So stop share.
All right, and I will start to share. And I'm going to take a second and open up that chat, so hopefully I can keep up in real time. I'm teaching all online this semester, so super exciting. And yeah, I feel like we're all whiz pros, trial by fire at this point. Thank you so much for that kind introduction, Carrie. And I am also really happy to finally be seeing the fruits of our emotional and logistical labors last semester. I hope all of you are doing well in Florida. I hope to come see you in Florida someday. I always put up this XK3D, XKCD comic to start off these classes or lecture about the gut microbiome. One, as a plug for XKCD comics. I consistently laugh as much as I learn when I read these comics, but also it just always feels like a nice, almost like a cultural zeitgeist pat on the back whenever gut microbes are highlighted. And it's happening more and more. So, you know, start off with a little humor. And speaking of, you know, thanking people for team efforts, it's not just about me and my gut microbiome. I always try to start off talks so I don't forget at the end, like, thank you. This is not just my work. This is, you know, really excellent collaborations across institutions, across disciplines, across specialties. So a lot of awesome people who's have all contributed to the research I'll be talking about here. So I'd say with that kind of out of the way, I'll say gut microbes are my first and truest love, but I also love making and eating food. And fermented foods are a model for what happens in our guts, right? And importantly, I've found out more recently, they're much easier to take into classrooms than poop and guts are. So, you know, I'm Learning that this has enabled me to engage the general public and students in middle schools, high schools, and undergraduate classrooms to learn about microbiology by collecting novel data. So I have been invoking increasingly citizen science and fermented foods as counter to science. Particularly in a pandemic, it's very useful to, make things relevant in a positive light where microbes are concerned in an age of viral transmission and germs. So I'm finding everybody eats, everybody poops. It's not glamorous, but it does make my work immediately relevant on a daily basis. So, general disclaimer, broader impacts and all of that. Now, just take a moment. If you want, you can just look at your hands, consider you are covered in microbes, right? You're outnumbered 100 to 1 on a cellular level. And genetically, genomically, you are more microbial than you are human. You're a super organism, right? So you're covered in microbes inside and out. And these microbes are involved in all of our life processes. One might say from cradle to grave, right? From development in early life to digesting our foods and promoting good or bad health, and even altering our behaviors through the metabolic products that these microbes produce when they digest our food. They're not just nutrients for us, they're chemical signals that can even cross the blood-brain barrier, right? So if you think about it, our guts are hugely important. Our guts are in fact our primary interface with the outside world around us. Even more so than, we might think that our skin is our primary interface. Your gut is your body's way of learning about the world. So let's take a quick tour of the gut to get you oriented, right? Food contains starches, proteins, fats, and fiber. So I think about food as these 4 main categories of macronutrients. And our bodies digest those different nutrients at different sites along the gut. As soon as we take a bite of food, amylase enzymes in our saliva break starch down into sugar. And then as food travels through our gut, proteins are digested in the stomach and fats are digested in the small intestine, right? By the time our food gets to the hindgut, fiber is pretty much all that's left. And lucky for us, the cecum, which I lovingly call a wonder pouch, and the colon are filled with gut microbes that digest that fiber for us. In fact, our gut microbes digest fiber so well that we animals never evolved this ability on our own. We are completely dependent on our gut microbes to digest all dietary fiber. So take a second, digest that, if you will. That essentially means that herbivores couldn't exist without gut microbes. Camels, manatees, hippos, deer, sheep, cows, horses, giraffes, kangaroos, rhinos, vegans, right? You name it. If they eat plants, they couldn't exist without gut microbes. We have a lot to be thankful for. So previous studies have established that gut microbes vary depending on diet and on gut morphology, right? So here you can see a principal coordinates plot that graphs the similarity between microbial communities that were isolated from 116 different animals belonging to 56 different mammalian species, right? So in this plot, each dot represents an entire gut microbiome, right? So that could be, we now know up to, 1,000 or more thousands of species of bacteria, right? Up to 100 trillion microbes or so, give or take. So on these plots, the closer 2 points are, the more similar the microbes within that community are, right? And we can color those dots to identify patterns based on different animal characteristics. So in this panel A on the left, you can see that communities cluster based on diet, right? Except for bears. Bears are weird. We'll come back to them in a bit. And then in the panel on the right, we can recolor the same samples based on gut type to show that animals evolve different gut shapes or different gut types or complexities of morphology to digest their different diets, right? Carnivores and omnivores tend to have simpler guts because we can digest most of the food ourselves, right? We're really good as animals at digesting fats, proteins, starches all by ourselves. But herbivores eat lots of fiber, so they depend on gut microbes to digest their food, right? So they've evolved longer and more complex guts, both to provide more space for more microbes and to give those microbes more time to digest all of that fiber through fermentation, right? So Lay et al. Were the first researchers to really map out these widespread patterns across species in 2008. But in grad school, I wanted to know all about these communities, more about how they form, the process of succession, right? How they vary across different species, diets, and gut types. Okay, so again, super glamorous. To do this, I collected hundreds of fecal samples from, and I've lost count of the number of species at this point. I think it's well over 20 at 4 different captive institutions. And now I'm adding wild studies. So from those feces, I put them on dry ice or pop them into a -80 freezer immediately. In a pinch or in the field, we plop them into ethanol, right? Just to stop those microbes from replicating anymore, because we want a snapshot of what's happening in the gut. So feces is a great non-invasive way to find out what's happening in the gut. From those feces back in the lab, I extracted the DNA, and then I used different approaches. I either used 16S sequencing to target a 200 base pair region. I call it a genetic fingerprint, right? That helps me to identify which bacteria at the genus level are living in those guts at the time that the animal voided a sample, right? Let's euphemize poop. So 16S sequencing gives me the identity. It's analogous to reading the last names in a phone book, right? From those same DNA extractions, I could also use shotgun DNA sequencing. That's like kind of chopping up and looking at snippets of all of the resumes for all of the members of a community, right? I'm looking at all the possible functions that the entire community could collectively perform at a given point. And then I also then returned to those fecal samples and used NMR spectroscopy to measure the actual fermentation products, right? So in this case, I'm really interested in short chain fatty acids, which are what microbes are digesting fiber to produce. Short chain fatty acids are, butyrate can be anti-inflammatory. A lot of these short chain fatty acids are the nutrients that we can actually use, for our energy currency. They're kind of the important trait of our symbiotic relationships with gut microbes, right? And to continue this analogy, where shotgun DNA sequencing would be kind of like reading through the collective resumes, NMR spectroscopy kind of tells us what jobs the community is actively performing at any given time. In the same way that you and I have jobs that pull on some of our skills, but not all of our skills necessarily all at once, all the time, right? So the analogy is set. You may wonder, Duke Lemur Center, that seems captive. You mentioned zoos. Why captive, right? I really appreciated when I was first starting grad school that nobody had really mapped any colonization studies in non-human species yet, or if they had, they were of mice under highly controlled situations. We really didn't know a ton about these different processes governing and shaping and driving microbial community structures and diversity. So I love the fact that we have dietary control, but that at the Duke Lemur Center, they are still feeding lemurs, for the most part, a lot of food items that we as Western humans would be buying in a supermarket, right? Except for the primate chow. But everything else was, pretty common between us. So it provides a decent nutritional model. We have known life histories. We know exactly what day these animals are born, when they start eating solid foods for the first time, right? And similarly, we have highly detailed health records, which came into play for a future study, right? So I'm going to present a series of kind of case studies to summarize some of the main findings and questions that I was asking. First, I wanted to know about primary colonization. How do these communities form from the day that a lemur, in this instance, an animal is born, until they've reached sexual maturity, right? So when they're adults. And I compared this, basing it on the classic ecological literature for vegetation succession, right? So thinking about, how do we grow a forest from scratch? Do we see similar predictable processes of assembly in lemur guts? And to do this, I compared 3 different species of lemurs, 2 of which are fed a very similar fruit and vegetable mixture in captivity. But you can see they have a very different gut structure. Our red-ruffed lemurs in the wild have evolved to eat a fruit diet that's very high in fiber or in sugar and starch, whereas our ring-tailed lemurs have a more complex gut for a more omnivorous generalist diet. So we also have, kind of bang for your buck in the captive scenario, a common garden experiment of sorts, where we have the same diet fed to 2 species with different guts. And then you've got shafoks that eat leafy greens all the way, and peanuts. They love peanuts, and they have really long guts. They're kind of the cows or the goats of lemurland. And I sampled all of these baby lemurs across their adorable lifespans at 6 different developmental time points. And what I found is that these communities do assemble predictably, right? When I was measuring the alpha diversity, in this case, Shin and Weaver diversity is a measure of relative complexity of communities that takes into account both the number of different types of organisms, or the richness that are present in these microbial communities, as well as their relative abundance. So some of that community structure, right? Found that in general, diversity, the communities get more complex the older a lemur gets, but a fiber-fed lemur, right, the shafoks with the long guts and lots of fiber, are able to host the highest diversity of bacteria, right? It's the most niche space. It's the most stable environment. That could be your climax hardwood forest, right? But then if we think about beta diversity, or the difference between the variation between individuals, we see that within each species, beta diversity is actually decreasing. So even as these communities become more complex over time, they're kind of honing in on a stable and consistent adult microbiome. They're growing the perfect preferred hardwood forest for life in a shafak that eats leaves, or a ruffed lemur or ring-tailed lemur that eats fruits and vegetables. And you'll notice that just as the shafak has the most complex communities, they also have the most consistent, which is suggesting to me that fiber is highly selective. So you have a lot of fiber digesting bacteria, but you have very specific lineages of bacteria that digest fiber, right? And we see that even more clearly when we look at a principal coordinate plot. So again, each of these dots represents a fecal sample microbiome from an individual at a different life stage. That's the different shapes. So moving right to left, I've now learned from data visualization that I should have flipped this horizontally so we could progress from left to right. But in this case, just flip your brain for a second, right? Our youngest baby lemur samples are on the right side. They're colored by the different species. And then moving from right to left, you see those animals' microbiomes are developing even as the individuals themselves develop, right? And I've put these circles or ellipses around all of the samples collected from animals that were only eating solid foods. So after they've weaned, we see kind of the stable, mature climax community cloud of microbiome, right? What does it look like for a normal, healthy adult or nearly adult lemur? And we can see, again, several patterns and variation, right? You see a lot more variation in animals that are eating more easily digestible or less selective diets, right? Those ruffed lemurs and the ring-tailed lemurs eating the fruit and veg mix. So you see a lot of a lot of variation between individuals. So their diets and their guts are not as selective for specific types of microbes, but you also see some overlap that I think again is a nod to that common diet they're fed in captivity. By contrast, right there in the center of the bottom, you see this really tiny little fistful of green spots, and that's all of your leaf-eating lemur samples. Pretty much as soon as they have their first taste of solid food, the fiber diet is so selective, coupled with that really long GI tract, they just have, they're selecting for a highly specific gut microbiome. To eat leaves is to be an adult for a shafak. And just for kicks, let's see, I'm going to turn on the annotation tool and show you where my microbiome landed, because you have to look at these things. Here I am, that star that I just stamped. Eating fiber can make even a human with a ring-tailed looking gut actually shift toward a really leaf-eating lemur, a very long gut morphology. So you can compensate for your gut limitations through diet if you so choose, right? The power of lentils and salad. So that's the big takeaway there, right? And now you know more about me than a lot of people do. So I also wanted to make sure, again, so far I've been talking to you about the amplicon sequencing. So we're talking about last names in a phone book. Now I'm going to give you that reading the resumes of these collective microbes, right? So I was trying to figure out with shotgun sequencing, I was able to actually identify start and stop codons for full gene regions, right? That then code for all of these different putative functions or enzymes, right? So using that shotgun sequencing, I'm trying to figure out how do I even visualize to you the tangled snarl of metabolic pathways. Google keg diagram sometime, and it just looks like a ball of rainbow yarn. And I thought, well, really, it's kind of like a subway diagram, right? I mean, you're bringing in your food, your different nutrients are almost getting off at different stops along the gut. And that's all thanks to the different microbes and the enzymes that their genomes encode. So I looked up blogs on how to make subway diagrams, and here you go. Here's a metabolic pathway subway map. And you can see, the orange circle line is your amino acid digestion. You've got the green line at the bottom is your secondary metabolites. That central purple, like the spine of the metabolic subway, is the TCA cycle, right? Moving south toward, our central energy currency, right? So this metabolic pathway subway map includes all of the stops used by all of the microbes in fruit-eating and leaf-eating lemurs, okay? So your gamma pool, if you will, every possible stop is there. Now, when I zoomed in and looked only at the microbes in the lemurs eating fruits and vegetables, I found only a subset of the stations are in use. They're not eating a lot of fiber, so they don't really need to know how to use like chlorophyll from leaves, right? You don't see the green line at all. Compared to the leaf-eating lemurs, they don't have much use for sugars because the lemurs aren't eating a lot of sugars, right? So that was really interesting to see, this segregation of the metabolic subway map based on diet, right? So moving from there, I was then really curious about these extreme herbivores, right? So again, in general, when we think about patterns in gut morphology across different mammalian species, we tend to find that carnivores, because they're eating easily digestible proteins and fats, they tend to have really short guts, right? Almost just a garden hose, only like 3 to 6 times the length of their body. Then at the other end of this spectrum, we have, 16 times the length of their body, the shafaks eating all the fiber. So there's a bamboo lemur, there's a red panda, and a giant panda, all of which, whether you are at the Duke Lemur Center in Durham, North Carolina, or whether you're at the Smithsonian, the National Zoo in Washington, D.C., these 3 species, by happenstance and luck, are, but also by design and nutritional responsibility, right, are all fed essentially the exact same diet. They're fed 95% bamboo, which is just hugely fibrous, very tough to eat, and sweet potato treats. So based on all of that fiber, one might expect their guts to be way down, on the 16 to 20 times the length of their body end of the spectrum. But in fact, their guts are only 3 to 4 times the length of their body. This is a huge mismatch, right? How are they even living? No wonder they move so slowly, right? It turns out giant pandas are likely compensating for that really rapid transit time with a very slow metabolic rate. Don't burn energy if you're not taking in very much energy. And they're like cows, they just eat all the time. But they've been a mystery in the microbial world for over a decade. People have been trying to figure out how are these fiber digesters with garden hose guts actually surviving on their diets? What is going on? So I thought, oh, I can do this. I can figure out what is going on. So I compared those 3 bamboo specialists to the ring-tailed lemur, which is a sister species to the bamboo lemur. They're only about 13 million years diverged, which sounds like a ton, but that's kind of like humans and chimps, right? Close cousins or sister species. Compared to the bamboo eating carnivores, those rogue carnivores, they are 60 million years diverged, and their last most recent common ancestor with primates was 83 million years ago. So dinosaurs were literally still roaming the earth. Okay, so just for context, this gives us a really great system for thinking about, is it who you are in your evolutionary history, or is it what you eat? What's the most important driver? And as with most microbial stories, the answer is both, kind of. So on the one hand, phylogeny exerts huge influence on the community as a whole. The most dominant microbes tend to differ between host species, right? So in this principal coordinate cluster, you can see we have, all the dots that are the same cluster or the same colors are the same species, right? So you see a very large driving species effect. And then I added these gray circles that represent the top 10 most abundant types of bacteria that I identified in these data. So you can see giant pandas, 90% of their gut is Enterobacteriaceae, like a generalist, possibly a pathogen, but certainly something that's just in and out so fast that that was a good snap, that it just doesn't have time to make the panda sick, but it's also really good at taking advantage of whatever it has while it's there. Whereas the lemurs are actually, with those longer guts and that closer relatedness, they're connected by many more microbes, right? So they're more closely related, not only evolutionarily, but also from a microbial perspective, okay? But when I zoomed in and said, forget about the evolutionary history of the microbes, how many microbial genera, right, specific types of microbes are shared between each of these different groups? What I found is that the 3 bamboo specialists share many more microbes than we would expect by chance. So there were 48 different types of bacteria that were shared across these 3 very distantly related. Remember, dinosaurs roam the earth, right? Compared to only, 8 types of bacteria that were shared by the sister species. So you have 6 times more shared types of bacteria just because of that dietary driver. Eating fiber is a powerful thing, right? It just goes to show, again, how powerful a fiber-rich diet can be in shaping the gut microbiome. Okay, so now I'm going to shift. We've talked about healthy stuff going on, but people always, there's like a sick fascination with being sick, right? So we've talked about primary colonization. What happens when you chop down the forest? Can you regrow the same forest again, right? So that's a story of secondary colonization. And lucky for us, unlucky for the lemurs, having a really long gut actually makes you hugely vulnerable and susceptible to cryptosporidiosis, which is when these shafaks, as soon as They have weaned from their mothers and they go outside into Duke Forest for the first time. There's a seasonal outbreak of cryptosporidium. It's in the environment. All the lemur species are exposed to it, but the shafaks have a long enough gut that they actually get sick. So every year, most every year we have outbreaks. So a great thing about this is the same individuals that I had studied their primary succession from birth to weaning got sick. So I was actually able to compare their secondary colonization from when they got sick to when they recovered. So again, a longitudinal sampling strategy, right? And here's kind of that sampling map. The main takeaways here are you have different lemurs are affected differently by the same illness. And part of this has to do with how old those lemurs are. Gisela and Remus were sick for a very long time and they had very intensive infections. They were brand newly weaned, right? They were the youngest animals. Beatrice, on the other hand, got sampled way retroactively because she was asymptomatic. They actually, looked at her feces by chance at the end of an outbreak just to see who else was there, and she had oocysts and just didn't matter. So different age groups are affected differently. Yeah, so there you go. But we were able to sample pre-infection, during infection for everyone except Beatrice, and then we were able to map their recovery over time. And so again, looking at the Shannon index, that alpha diversity, the measure of complexity, how many types of bacteria are there, you see there's the healthy bit, right? Usually healthy lemurs have a Shannon index of 8 to 9 that crashes as soon as they are actually experiencing a washout of their native microbes, right? That would be euphemism for extreme terrible chronic diarrhea. Euphemisms preferred usually. But then you can see they have a slow recovery period back to a stable point that is similar to before their primary infection, right? And we see the opposite pattern when we look at the beta diversity, which again is, in this case, it's a comparison of every lemur to themselves before they got sick. So your pre-infection difference from your healthy self is 0, right? So what we see here is the sicker you get, even though you are losing more and more of your original healthy microbiome, you are wandering way farther and farther away from your healthy baseline. Okay, so what you want from a unifrac distance perspective is to get back to 0, right? So we see that even 65 days after primary infection, they're not quite back to where they were to start with, but they're getting there slowly, right? So I won't go into much more detail there, but I was able to show that disease predictably affects diversity, right? So as kind of a recap, because I think in pictures and I've found that sometimes that helps for other folks too, we actually see a brief increase in diversity at the beginning of illness, and that's likely invasion by those opportunistic pathogens, because even for an animal with a very long gut, diarrhea just speeds things up, and that takes away your selective power to regulate your gut microbiome. Then we see that native washout and the use of antimicrobials, right, which would certainly be the case for treatment of human illness. We see the crash and burn of the original microbiome. And then we see a slow recovery, but eventually regaining stability, as we see in that kind of plateau, right? So the next case, we actually had another unfortunate occurrence at the Duke Lemur Center that provided a wonderful opportunity to actually peek inside the gut, which we would normally never, ever do with endangered species, right? Particularly if they're in good health. But we had this opportunity to see what different communities reside at different sites along the gut. And this is definitely making the best of a bad situation. We had successfully fed avocados to lemurs at the Lemur Center since the early 90s, like 1990s. But in 2016, we had 4 lemurs stricken with person toxicity. And within 24 hours, we had lost 4 otherwise healthy, previously healthy eyes. So this is my disclaimer, blood and guts. So if you do not want to see what the guts of a lemur look like, just cover your face or turn away, and I'll let you know when it's down. So from these lemurs, I had already been dissecting lemurs that were euthanized for health issues for years. So when we heard that 4 animals were being euthanized or were dying of this toxicity, I said, you know, I'm here. You seem swamped. Let's do this. So it was a once in a lifetime, I hope, opportunity. But this is what the gut of an eye looks like. A really exciting thing here to take note of is that the eye has an appendix, right? That little worm, pinky looking thing coming off of the wonder pouch, that's an appendix, just like you and I have. So again, a really exciting opportunity to see how microbial dynamics in lemurs might model humans, right? So from that, we then took gut samples from the cecum, from the upper colon, and then from the rectum right before the gut content becomes feces, right? So what you see first from this otherwise messy looking bar chart, it looks fairly consistent to the naked eye across the GI tract, right? Which I am learning now from more and more studies. In animals with a cecum or an appendix, that is a perfect, it's like an oxbow lake. It's a slow down eddy pool. So in animals that have a cecum, their gut microbiome really seems to stabilize in the lower gut. You see a lot more consistency there. But what was strange about the eye is that we saw over half of the microbiome in the lower gut was Prevotella. Now, that's a specific genus of bacteria that's really good at digesting carbohydrates, right? And Prevotella have actually co-evolved with primates for a really long time. But really interesting, I hadn't seen a species that had so much Prevotella before. And that may have a lot to do with the fact that eyes eat a lot of insects and nuts. So they eat these really high energy, structurally defended food items. So then we wanted to know, you know, how does the microbiome vary along the gut? And when we compared the alpha diversity, we actually see there is less complexity in the appendix compared to the cecum and colon, but that those fewer types of bacteria are more evenly distributed. So you have a more even representation across the community. So really interesting. What is in the appendix all does really well, right? You don't have some stragglers that are just rare members of those communities. But once you get past the appendix, the cecum and the colon look very similar. So it's all a mixing vat, right? And what was really exciting about that, we also see a lot less variation, you know, when we measure with beta diversity and unifrac. So less variation, but a lot and much more specific communities. So when we wondered, you know, what makes the appendix so different, and we looked at the specific types of microbes in those communities, we found Methanobrevibacter, which produces methane. That's really important for driving digestive efficiency. So that's kind of what you would want in your oxbow lake, right? We saw Akkermansia, which produces anti-inflammatory compounds. That's something that you would definitely want sitting in your appendix because it's a lot of immune tissues, right? So interacting very intimately with your immune system. And together, this was really exciting because it upheld some theories that have been presented by Bollinger et al. And later also supported and by Smith et al. This idea that the appendix might be a safe house for beneficial microbes, right? So that after an infection that, you know, results in a native washout of the bacteria in your gut, then you might have this safe harbor of microbes that can then repopulate the gut. So those are my primate stories for now. Now I'm going to transition from captive studies to a more recent shift from primates to carnivores and in wild populations. So on the left here is Sierra Gilman, who recently graduated with her master's from Northern Michigan University and is now getting a PhD in Washington. And on the right is my collaborator and Sierra's master's mentor, Diana Lafferty at Northern Michigan University. So they're with a black bear. we were really interested, if we see these differences between different gut sites in primates that have a cecum, what about black bears? And they're in the Upper Peninsula in Michigan. So they actually engaged hunters of black bears as citizen scientists. And this was a great opportunity not only to engage a demographic or a population of people who maybe aren't always involved in science, but also who really believe in their baits. They, like the cheese man, which is what we call him, right? He honestly believes that his cheese baits are going to get him the best bears every time. Then there's the hunter who wants to go all natural and only gives nuts and berries, right? So trail mix, but without the chocolate. Then there's the saltines and Skittles guy, right? So there is a wide variety of baits that are used. So we're able to actually think about not only differences in diet, but also differences in anthropogenic feeding behaviors, right? So we, again, sampled from the jejunum, so the small intestine, and we compared it to the large intestine, right? We didn't have a cecum 2 sample. So what we did was give them different kits with 2 standardized lengths of string. We said, go down this far from the stomach and then go up this far from the anus, right? So we knew that we were getting the same region of the gut every time. And you can see in this bar chart at the genus level, there are a few genera that seem to be different, but statistically, the jejunum and colon really don't look different at all. So if you have a garden hose for a gut without a cecum, you also really don't have differentiation, right? And we also found that a lot of these different types of bacteria in humans are associated with obesity, which is bad news for a human typically, but it might be actually a really good news for a black bear in a place where they're still hibernating. They're not all hibernating in North Carolina, right? And Florida, I doubt it. So that these anthropogenic foods, these hunters' baits might actually be benefiting the bears if they need to fatten up rapidly in order to hibernate. So some really interesting implications for management. And then when we looked at the proportional contribution of corn to the diet, we found more implications that maybe have something to say, can help us reflect on our lifestyles as humans, right? We found a significant correlation. The more corn you eat, the less diverse your gut microbiome. So if you want to have higher complexity, then eat less corn, right? So to recap, what have we learned? We've learned that gut microbes complement host diet, even across vast evolutionary distance and even at different gut digestive sites, right? But we've also learned that short guts are a disadvantage if you want some sort of differentiation, or if you want the chance to regulate your microbiome, to select for specific, more beneficial species of bacteria. So that's what we've learned. What can we do with that knowledge? We could eat more fiber if we want to select for more complex and more beneficial microbes living in our guts, right? Producing more anti-inflammatory compounds, perhaps. We might want to avoid sugar and processed foods. Maybe avoid corn for, a ton of our diet. And also we can compensate for our relatively short guts with fermented foods. So if you can't expand the length of your gut, right, you can pre-digest your food in a jar. So, that's maybe not the most appetizing way to think about lactic acid fermented foods, but if you think about it, This is one of my home ferments. So it's carrots, bell peppers, cauliflower, and then there was a lot of garlic because I'm a garlic fiend. So when we pack produce into a jar and add, a light saline solution, environmental microbes settle in, but the salt and the low oxygen environment are selective, right? And they select specifically for lactic acid bacteria that then digest the starches, the carbohydrates in that produce to digest carbon dioxide, which is why you might want to burp your jar a couple times, or why it'll hiss when you open it for the first time. So they're producing carbon dioxide, which further decreases the amount of oxygen there, right? And it makes it even more of a gut-like environment. And they also produce lactic acid, which drops the pH below the threshold tolerance of a lot of food spoilage microorganisms, including molds, right? And while they're doing that, they're also producing, some vitamins, some short chain fatty acids. They're producing things that our body, that both increase the shelf life of the food and increase the nutritional value of the food, right? So even though we might not have a ton of time to digest that food, it'll be more readily available for us if we eat fermented foods. And sourdough starters are also incredibly compelling and provide another way to ferment food before we eat it. I think it's really interesting to think about, what a staple bread is in daily life and also across human cultures, right? But if you tried to eat flour on its own, you would starve because plants don't want you to eat their babies, right? The seeds that we grind into flour are, the babies of plants. So to protect them, plants are actually producing a lot of phytic acid and other compounds that bind nutrients and that bind trace minerals. So if you can't beat them, starve them out is the plant's evolutionary strategy, right? But by fermenting a flour and water mixture, you're actually giving microbes time to digest those different compounds. So you're unlocking nutrients, right? And flavor. And this is a part where I get to say, I get asked all the time whether sourdough is a probiotic. So I just want to take a moment to clarify things, right? Sourdough is a prebiotic. Whole grains especially are full of fiber and other compounds that help fuel a healthy gut microbiome, as you know. Probiotics are foods that contain living organisms. So probiotics would be things like yogurt, kimchi, sauerkraut, kombucha, right? But while sourdough starters are chock full of thriving organisms before we bake them, the baking process actually kills everything in the starter before you consume it, right? So fermented foods, the produce in a jar, that you're fermenting, those are live. Those are your probiotics. And they contain still some fiber and other carbohydrates. So fermented foods are all prebiotics, right? But sourdough, even though it's a dead food by the time we're eating it as bread, before they die, those microbes in the sourdough starters work incredible fermentation magic, right? They synthesize, or alchemy, if you prefer, right? They're synthesizing vitamins, they decrease the glycemic index, and they even digest gluten, right? Though to this point, there's some evidence that gluten-sensitive consumers react less to sourdough than to bread that's made with baker's yeast, but I would not guarantee anything, and it certainly doesn't apply to celiacs, right? So my disclaimers are out of the way. So I guess with those definitions, I'll just end by reiterating that, I've learned a lot about guts studying guts. It's been a really exciting journey thinking about these invisible multitudes that live on and inside of us, learning how to leverage what we learn from wildlife, both in captive and in free-ranging populations across species, and then figuring out how to leverage that knowledge for our own lifestyles so that we can, enhance our own health and well-being. So with that, deep breath. Thank you so much for your attention, and I'd love to open it up for questions.
Oh, that was fascinating. I loved it. So I have the chat open for anybody out there that wants to post some questions. please type your question in.
And Carrie,
I have,
to answer your question about the bears are all free ranging, so they eat whatever they can find, and they actually will visit, you know, any bait pile. They're also eating berries and, small vertebrates and bugs. I mean, the bears are just doing bear things. And yeah, at the point where they're shot by the hunters, it's completely opportunistic. So we tried to gauge what they had been eating, by measuring the stable isotopes, carbon and nitrogen, from their, I think, guard hair samples. So, and I see, Barry, you said, any suggestions for those of us who no longer have their appendix? That is a great question. I would lean personally even more heavily on eating prebiotic and probiotic foods, because you won't have, I'm assuming it's you, I'll talk to you like you're the one, asking for a friend without an appendix. If you don't have an appendix, then you're lacking that safe haven. So you would be at greater risk, or I would predict that it would take you longer to recover after a washing out event, right? So I would lean more heavily on eating a fiber-rich diet, of avoiding processed foods. If you're not eating fermented foods now, maybe this is your motivation to start. Yeah.
so these studies have impacted your diet, or have you been interested in these studies because of your relationship with food?
Yes and yes. So I started my PhD in 2011, and I have a 2-year-old now, but before I was eating for 2, I was, eating for 100 trillion, right? As I was doing lit review, as I was gathering my own data, I was also burning the candle at both ends. So I was exhausted all the time. I commuted between cities for my 6-year PhD program. And then I was trying to have a full active social life. Yeah, in the strive for balance, I just stopped sleeping kind of. And I felt exhausted all the time. So I was trying to think of, when do I feel really bad? It's if I am eating a lot of processed foods, or if I'm eating a lot of, high fat or high sugar starch foods, the most delicious things. Um, and so I started to eat smaller portions, like snack throughout the day. And I started eating like a wilted kale salad with walnuts, goat cheese, and lactic acid fermented beets on top. That's how I got into fermenting. I wanted something tangy and crisp on my kale salad. And then I was hooked, and my kitchen is live now. I I lacto ferment pickles, and I've had a few. When I had kombucha, I would have scobies growing in the necks of my jars. So you'd take the lid off and take the plug out, and then you could eat what was pickled inside. And I actually did find, I started out just in the blind faith of like, I've sequenced these microbes and I've seen the results. So I want to go ahead and, select for things that I know from the literature to be beneficial, but I actually felt a lot more alert. So now it's a lifestyle choice, and I'm more motivated now that I'm teaching someone else to eat solid foods as well.
We have a question from Carol.
Hmm?
We have a question from Carol. Can you read that?
Yeah, Carol, you said, any plants better to use for fermenting to increase the diversity of good bacteria? That's a great question. I assume you're asking about lacto fermenting, right? I think in general, if I'm eating an unfermented food, then the more fiber and the less sugar, then the more selective it will be. But if you're doing lactic acid fermentation, then by adding a bit of salt and by giving it time to ferment, you are growing beneficial microbial communities either way. No matter, I've fermented apples, and I've done blueberries and blackberries. I've also done beets. I love sauerkraut. I I make purple cabbage sauerkraut because I love the color. And then I think, oh, brightly colored foods have vitamins in them. So I'm getting a twofer, right? Um, Carrots are great. I I haven't studied whether there are differences in like the total bacterial load for probiotics that are fermented from one produce versus another. I just tend to trust that if it's fizzy and tart, you're good.
I was typing a question, but I'll go ahead and ask it right now. How do you reach out to middle school students and younger audiences with your work?
That's a great question. Let's see. I have some, I'm trying to see. Here is a picture of one of the teachers at a local middle school. So um, throughout grad school, while I was waiting for the lemurs to grow up, there was a little bit of lag time before I could actually start sequencing the DNA in the fecal samples. So while I was waiting for the, to complete my sample collection before sequencing, I did a lot of outreach because I love telling people the stories of science. To me, I just want to tell you about what I'm excited about. And if I can't tell you in a way that you understand and appreciate, then I can't function as an extrovert in the world, right? So I was looking for all these outreach opportunities, and I don't know how I found the first ones, but it's like once you find one middle school event or elementary school event, then you tap into this whole network of educators who are really eager to have guest scientists. Um, so I've been really lucky to find those networks. I know there are a lot of local and national networks of educators and of outreach programs. Here in Raleigh, we so Rob Dunn, who you discovered me through, he had written an NSF grant that got funded that paid for my postdoc that he called Students Discover. So the whole point of that was to engage citizen scientists and the general public in novel discovery through data collection. So, the Global Sourdough Project, which I'm excited to say is finally out in the world. It's been published, y'all. It's in eLife, and you can find it. And we've just put up a beta test of like the big public facing. You saw the figures in the paper. Well, now you can see your own sourdough samples in our results. So I digress. You find one and then you hear about more and more through word of mouth. And I've also been really lucky to tap into networks of instructors, of educators who are eager to engage with the science and who are also willing to give me time in their class, not just as a 30-minute talking head, but developing citizen science protocols. So this picture is actually from a study of sourdough succession. I was able to pay this is Kristen Kemp and 3 or 4 other middle school teachers in Raleigh to, do science over the summer. So teachers got paid during the summer. So that felt like, I don't know, almost a justice mission for me. Public educators, right? But then also to engage them in the science so then they felt more confident in teaching science to their students. And part of this effort, we have a publication that's under review, like mapping colonization in starters. So, that enables us to tap into partnerships with local bakeries or not local bakeries, right? International baking communities, and check in on, all of the anecdotal wisdom that has been passed down for millennia, right? We've been baking bread for tens of thousands of years, but nobody's ever really checked in to say, when bakers say you can use a starter after 7 or 10 or 14 days, is that true? And how much variation is there? And how does that change flour type to flour type? And what bacteria are in there really? So we've got some of those answers coming out soon, hopefully, under review.
Yeah, that was a really cool study. I think I saw it on your Facebook page, and then I shared it on ours. Before we go any further with questions, I wanted to give a shout out to Tallahassee Scientific Society because I really failed to introduce the organization that is sponsoring this before we started tonight. And I'm really curious about some of the people whose names I see that are participating in this. I'm not familiar with your name. So if you're not a member of TSS, would you let me know through chat how you heard about this talk tonight? We're always trying to increase our outreach to the public. And so any different mechanisms that are working, I'd like to know about, whether it's Meetup or Facebook or word of mouth or FSU biology calendar. That would be very helpful.
And would you like me to stop sharing my screen?
Oh no, we like looking
Okay.
at Kristen.
Okay, awesome.
That's fine. Okay. Um, all right. So, did we miss a question?
Yeah, Nelson wanted to know, do you have any suggestions for buying off-the-shelf fermented foods? If you are looking for live probiotic foods, then I would look for live culture inside, right? So for a lot of kombuchas, for vinegars, there are some vinegars that are like, I think that even the 365 generic brand from Whole Foods is marketed with the mother in it. So, check that labeling out. If it says live inside, then it's alive. But yeah, that would be for probiotics. Yeah, otherwise it may have been pasteurized and things will be prebiotic. They'll be great nutritionally. They'll just be dead. And then Rebecca, awesome. Thanks for participating in the sourdough project. My starter's name is David Dowie. It only took me, 4 years to think of the name, but I think it was worth the wait.
That's great.
Can I ask how this teacher then used this in her classroom?
Yeah. So let's see, in conjunction with this summer-long research project, I also secured funding for us to have several sessions to develop an entire teaching packet. So on the Sourdough for Science webpage,
Sorry, we lost our audio there for a bit.
Yeah, I think she just froze. Her picture's frozen also.
Okay. Well, I don't know. Hopefully she will unfreeze.
Yeah. Or she'll
But-
rejoin, maybe rejoin.
Okay, because we have a question about wastewater treatment plants. So
Maybe she's fermenting.
all right, she's taken that off. And she looks like she's gone, and maybe she'll join again.
I bet she will.
Weren't we supposed to quit at 7 anyway?
I thought it was 7:30.
I had left it open until 7:30, but
Yeah.
obviously just depends how long we're going. And let's see.
Well, this was great. Thank you.
Well, yeah, I really appreciated her talk and see that she could have split that up and had it purely gut bacteria analysis or fermentation. I think next time we'd like to maybe include some more food scientists and have her talk about the science of fermentation and the role that has played in development of cultures and peoples over time.
That would be lovely.
Yeah, that would be great. So, well, maybe something else happened to her at 7. I haven't received a note from her. So I guess we will end it there. Again, this was Tallahassee Scientific Society that offered this webinar as part of our Horizons program. And we are still putting that together for the rest of the season. It's been a strange year, don't you think? So we're struggling a little bit with that, but I appreciate everybody showing up, and spread the word about TSS if you know people that might be interested in other talks or other outreach opportunities, etc. So check out our website or Facebook. See you guys another time.
Carrie,
Thank you. Thank you.
Thank
Oh, here
you.
she is.
hey, I'm back. Sorry.
Oh, there she is. Okay.
I had my internet blinked out, so I'm on
Yeah.
a mobile now,
That's,
but I'm here.
Sorry about that. We were saying our goodbyes, but there were a couple other questions in the chat session that if you could take a look at. One about wastewater treatment plants, and another one about
Let's
COVID.
see. Yeah, you'll have to, I lost the chat when I switched from computer to phone, but if you can read them aloud, I'll field them.
Okay, let me see if I can. Let's see.
Yeah, sorry about that. I'm glad it waited until I was done the talk.
Yeah, me too. And I'm glad at least a dozen of us are still here. So one question was about how COVID interacts with the gut, or what the gut does with COVID virus. What happens to viruses in the gut in general?
That's a great question. So Lawrence David, who is at Duke University, has actually posed a hypothesis that the gut is a giant mixing vat for viruses. That when you eat food, if you're eating raw foods, then plants have viruses, and if you perhaps take in some insects in your diet, whether you mean to or not, right? Those insects have viruses, and that then those viruses might all come into contact with each other in your gut. So I think that's pretty interesting. Also considering, the gut as a primary interface with the outside world, it might be a way to, in the same way that a lot of people say, eat pollen to help your allergies, because it's a way to come into contact with an allergen without, not through the respiratory system, right? It might be a way to educate your body about the world in a way that might be less of a flare-up, but I'm hesitant to say that with any confidence. I am no virologist. So and I could see that working in different ways for viruses that operate under different mechanisms of infection and transmission, right? So I'll try to pivot away now.
I'll pose another question here, but people are talking about COVID, so that was a very relevant question. All right, so this is another question. Can you talk about any implications for managed infrastructure systems like wastewater treatment plants, opportunities to learn more about public health, et cetera? I work with an educational facility that represents city utilities, and looking at incorporating microbiome ecology and how we benefit from microbes in anaerobic digesters and wastewater treatment plants, which that's a great question.
Yeah, absolutely. I think, so first, there's definitely a rich body of research on bioreactors, right? And on treatment plants. I haven't read a ton of it. I had been reading up on it in 2012 and 2013 because I was thinking, what if I can, have a highly experimental compost bin that then we could test priority effects of what happens if you start your compost with fruit versus vegetable, or, green versus brown material, if you're into gardening speak, right? But it's been a while since I've read up on that. I think the most use I would be answering that question at present would be thinking about ecologically, if these are anaerobic digesters, then what I know from the gut is that your fiber digesters, your leaf eating, your herbivores that take in a lot of fiber, tend to have anaerobic guts because they're a lot longer. Whereas we tend to see, so you're selecting for anaerobic fermentation with a long gut and with more fiber. Whereas animals that are bringing in more protein, more fat, more sugars, tend to have shorter guts, so they have more highly oxygenated environments. And you might expect more, like, say, sulfur reducing bacteria, right? Thus those stinkier poops from omnivores and carnivores. So I think it might be an interesting space to talk about the mismatch, right? What are we putting into our processing plants, and what are the environments that we're providing for that fermentation? Is an anaerobic digester actually the best way or the most efficient way to digest protein? I don't know. It seems counterintuitive from what I have seen in animals. So maybe there's a way to more accurately mimic the digestive processes and conditions that we see in evolved guts, if we want to make, biomimicry for efficient processing. I don't know.
Well, it makes for a great field trip for public schools to head there. And it sounds like he wants to provide some education for school kids.
Yeah.
I'm going to send you a message here. And I am probably going to close this out unless someone wants to ask one last question. I'm just so glad we finally got all this together.
Yeah, it's been fabulous. Thank you so much.
And the travel expenses really were pretty low for this. So thank you, Erin. Although next time I really would like to have you here in person. And
oh, that would be fab.
Yeah, I was saying while you were gone for a minute, having a session just with food scientists and maybe have a little lab project with some students in town, some K-12 students. But we'll talk about that another time.
That'd be awesome. Yeah, in December, I trained the North Carolina 4-H agents across the state for sourdough for the holidays. So I'm pretty well versed at this point in leading fermentation workshops. So.
Okay, that sounds great. Well, have a good rest of the week. And thanks again, Erin. And thanks everyone for showing up and being a part of it. See you next time.
Absolutely. Thank you.
Bye-bye.
Bye.